Good evening, everyone. We thank you for joining us tonight for the Biotech Pitch Club. I want to welcome those of you who this is your first time to attend, as well as those of you who are joining us. Some of you, I think we're up to double-digit pitch clubs, so welcome. And just for those of you who have not joined us before for Pitch Club, I just want to give you a little bit of information about AngelMD and then Pitch Club and why we do it. So AngelMD is a network of startups and clinicians, really with the focus of accelerating innovation in healthcare. This is such an important part for us to do, and so we have really focused on growing physicians and dentists that are interested in really through their time, and as well as helping drive healthcare forward. In addition to the clinicians and the startups, we do have investors of all sizes that are part of it, and so we welcome those of you who have attended tonight as well. The Pitch Club is really was originated as a way for us to drive engagement between the startups and the clinicians. And really, there's a few kind of key goals. One is giving exposure to these companies. We have a lot of companies on our network, and so for companies that have information they want to share and are in a great position to do it and are willing to do the work to present to you, they have the opportunity to come to Pitch Club. The second key thing is we fundamentally believe that clinicians should have a key voice in healthcare innovation. And so our goal is not only for you to be able to get access to this great innovation, but also for you to provide feedback through questions that you ask of the companies, as well as feedback through answering polls that we will make available to you after each of the presentations. So we do ask that you would participate in that and make your voice heard. It's companies to bring your knowledge into what they're doing, and it also ensures that your voice is being heard. And then the last thing is we really just want to have fun and be able to have education through the process of entrepreneurship. So one last thing, following the event, we do have time for networking. When you first joined, you might have seen there's tables. Each of our presenters tonight will have a table. So oftentimes we do not get through all of our Q&A. So if you want to have follow-up dialogue with them, you'll be able to join them at their table. And there's also opportunities to network and connect with other people who are attending the event tonight. So please do stay and join us for that. So with that, I am going to turn this over to Dr. Katie Richardson, who's our MC for the event.
Thank you, Jennifer. I am really excited tonight. Well, I always get excited about Pitch Club. And for those of you that have joined us tonight, we're excited to have you here as well. But we have three really great companies tonight. So it was a while back when we started kind of interacting with a few of these more biotech type companies that we decided we need to have a biotech and lab night, which is exactly what tonight is all about. And it is not going to disappoint. I will tell you, this is going to be really exciting. So our first company of the evening and speaker of the evening is Jim West, and he is the founder and CEO of a startup called Clara Biotech. One of the things, while Jim gets his slides up, one of the things that you're going to notice tonight, we do always like to hear about the science and the medicine surrounding these companies. Well, when you go into biotech, you're going to notice that a lot of these are very science heavy. So I'm going to say you're welcome in advance. And I know that this is going to generate a lot of questions from our audience. So Jim, go ahead and take it away.
Thank you, Katie. I'm Jim West, CEO of Clara Biotech, and we founded our company in 2018 to solve some of the major challenges and issues in the field of exosomes. And the goal of our company is to help move exosome technologies from the bench to the bedside. And this really represents an exciting and new opportunity to lay the groundwork for the next foundation of biotechnology. And let me start by telling you what an exosome is if you haven't heard of them before. Exosomes are the FedEx communication system of the body, and exosomes communicate and transmit cellular information like DNA, RNA, and proteins. They go between cells, and here's an image of one. There are about 100 nanometers in diameter, and there's about 1,000 exosomes to every cell in your body. They're prolific, and they have a number of really exciting and interesting applications in the field, starting now with delivery of RNA and nucleic acids to specific locations using the exosomes as a targeted delivery vehicle, but natural. Also, there's exciting applications in cancer immunotherapy where you can get the same benefits of CAR T-cell processes, but in an allogeneic platform where you can mass produce this product as a standard pharmaceutical. And a few years ago, we discovered that exosomes that come from stem cells inherit many of the same benefits and properties, including acting as an anti-inflammatory and promoting regeneration of tissues, but in an acellular platform. It also has exciting applications in the fields of biomarker identification, so liquid biopsy. Right up there next to circulating tumor cells and cell-free DNA, you have the ability to identify diseases like cancer, Alzheimer's disease, and other neurological conditions simply from body fluid samples. And the big challenge today is that most of the competing systems are based on some form of bulk isolation or density isolation that gives you a final product that is noisy with contaminants you don't want and can't get rid of. At Clara Biotech, what we've developed is a pure, active capture and release technology that gets exosomes and nothing else. And here's how it works. We start with an immunomagnetic bead, and to the surface we put targeted antibodies. These are totally customizable and tunable. We can put whatever combination we want on the surface, and it allows us to capture specific types of exosomes, tuning it to that specific need. Once it's bound to the surface, we can wash away all the non-exosomal content, and then we have a proprietary photo-release process that detaches the exosomes from the bead. And here's how it works. It's really simple. It works in any laboratory setting. It's based on immunomagnetic bead technology, which has been around for more than 40 years. So everybody knows how to put this in their workflow. You start off by mixing your sample, your body plasma, your urine, saliva, or cell culture media with our proprietary reagent. This is a single-use consumable. And once the exosomes are bound to the surface, you put it into the box you see here on column 2, and it releases the exosomes over a 20-minute period. The great thing about this platform is not only is it scalable and usable in any laboratory, but these exosomes are suitable for any downstream application, which is a really unique benefit in this field. Here's a comparison showing our technology to the gold standard, in this case, ultracentrifugation. With what you see here is on the left is a Clara bead with exosomes bound to the surface shown with the red arrows. And on the right is a solution of exosomes through ultracentrifugation, which shows a lot of noise and contamination. And what's really important is the graphs on the bottom. These are called nano tracking analysis reports, or NTA. And what you see on the x-axis is a size distribution from 0 to 300 nanometers. And on the y-axis, you have quantity. And so on the left with Clara's product, you see a narrow band of exosomes captured. And on the right, you get a much more noisy sample representing a lot of non-exosomal content. We can get highly functional exosomes too, and here you can see an assay using exosomes that have been taken up into dendritic monocytes. We fluorescently tag them, and you can see the signal that goes into this cell showing the green fluorescence. And we get highly functional exosomes relative to the gold standard. And this is really important to help solve future problems in manufacturing and scalable therapeutic grade exosomes. Like I said, we've been around since 2018. We started with an SBIR phase 1 from the National Cancer Institute. It's very competitive and very hard to get. We also completed what they call I-Corps at NIH, or do customer market validation at the very beginning. Shortly after that, we raised our pre-seed round to help get our exosome isolation lab service up and running and to move us forward into product development, which we're in the middle of today. In 2020, we started heavy development of our exosome isolation kit, and also participated in Mass Challenge Boston, where we were able to connect to the Boston ecosystem. From that, we were able to get a number of great investors and advisors from the community. And today we are more than 3/4 of the way through our seed round, which we're expecting to close in the coming weeks. And with all this in place, what we're doing right now is we're getting our first research use only exosome isolation kit to market. You can see it on the upper right of the screen. And this kit is something that we've got more than 10 beta sites around the world today using. And because of the goal of the company moving exosomes from the bench to the bedside, where we're going beyond the research kit is in supporting diagnostic applications for liquid biopsy and also solving serious problems around therapeutic manufacturing at scale. I founded the company 3 years ago. My background is in biomedical engineering. I also have a master's in entrepreneurship, and in 2016, I got one of the first mobile medical devices cleared by the FDA. My co-founder, Dr. Mehi, is the inventor of our platform. She's been developing this since 2012 and is an active researcher who's been published in Nature and other great journals. She's an expert in microfluidics, exosomes, and drug delivery. We also have Dr. Tom Kroll, a pharmaceutical drug developer who's helping us with pharmaceutical development and deal flow. Today, we have 5 full-time employees, 4 part-time employees, and we're actively sourcing for 3 new positions. Do you have any questions?
Jim, thank you. I invited our audience, but I will once again go ahead and put any questions that you have for Jim and Clara Biotech into the Q&A function. I'm going to start with a couple of my own, though. Again, I mentioned this at the beginning, right? This is pretty science heavy. For those of us that have been out of medical school for a little while, I loved the fact, by the way, that you reviewed what an exosome is for us, because many of us probably are thinking to ourselves, what is that again? But I'm guessing you talked about the use of this technology in therapeutics, and I'm guessing that purity is a really big, important piece of that. So talk about how your purity varies from the ultracentrifugation that you talked about.
Yeah, thank you, Katie. So the reason purity is so important and the challenges that the industry experiences with it today is when you do ultracentrifugation, you're effectively isolating based on a density gradient, or you're running it through a size filter and you're filtering based on size. And in both of these methods, we're talking about material that's 100 nanometers in diameter. And when you isolate this way, you end up with a lot of non-exosomal material. A lot of other stuff exists at this domain: cell debris, cell-free DNA, viruses, lots of stuff you don't really want. And so when you're looking at signals, you get a lower signal. You're looking for a needle in a haystack, and it gets masked. And when you talk about therapeutics, what the FDA and other regulatory bodies look for is pure, potent product. They want a clean, well-defined, active pharmaceutical ingredient. And today, with these filtration methods, it's very difficult to be able to cross that gap. And with our selective capture and release, being able to provide a pure, qualified active ingredient, we believe we can overcome those hurdles.
Which is why I'm guessing you're starting in the research market. Clearly, there's a need there for this type of technology.
The whole field's only 10 years old, so it's a very, very young field. And I don't have a slide for it, but last year in 2021, there were 5 $1 billion licensing deals between pharma companies and exosome manufacturers partnering to get RNAs and gene therapies delivered to specific locations. And so it's really heating up. It's a very active field. You are right. And from a company development perspective, you don't want to go straight to regulated manufacturing. You want to build up your way there. So we're starting in the research space, and we're going to grow that organically.
Fantastic. So Dr. Macaluso is in our audience tonight, and he has my second question right on the tip of his tongue as well. He puts it a slightly different way, but please discuss the major clinical application that you are looking toward. And I would say, tell us a little bit more about the most recent use cases. What are they using the technology for?
Yeah, thank you, Dr. Macaluso. So the use cases, as I mentioned, obviously delivery of drugs like a liposome or a lipid nanoparticle, that's that's coming, and there's products moving through the pipeline for those activities. What I get really excited for, though, are some of those other things I kind of mentioned. So cancer immunotherapy, I mean, it's huge that you can have a product that has the ability to train your immune system how to fight diseases, but without requiring like CAR T cell. One of the challenges there is you have a little bioreactor for each patient. So you give your own material, and then 2 weeks later you turn it around into a therapeutic. It's very difficult to scale that in the traditional biotech world. With our platform and with what we can provide, you can actually create a large-scale manufacturing process for that. I also think that it opens up the door to regenerative medicine in a traditional way. So right now, a lot of that technology exists outside of the FDA, and it works off-label. And I think that we have the opportunity to create actual labeled drugs for regenerative medicine.
Very cool. So another question from our audience. Talk a little bit about, you mentioned pharma companies, but talk a little bit about the main strategics that might embrace this type of biotech.
Yeah, and I think I didn't quite answer Dr. Macalusa's question there as well. So the strategics that we're looking for, it's really, we are a platform. We're building pickaxes and we're selling them to miners. We don't know who's going to hit gold, but they'll be using our pickaxe when they find it. And so we're really looking to be a catalyst for the industry. We want to support everybody in the work that they're developing. And so there's lots of opportunity and lots of applications that are being developed, and we want to work with and support them. And so today, we are working with a number of researchers, diagnostic and therapeutic folk. To be honest, our scale, we're still early. We haven't got our first product out. So we're still a ways away from therapeutic scale activities. But I can tell you that on the diagnostic side, we have interested companies in looking at biomarker analyses. And we also have some research use only applications with some major pharma companies looking at actually supporting clinical trials by looking at pharmacokinetic markers and other things like that exosomes can provide in a non-clinical setting, but allows us to actually have a scalable platform before we go to commercial clinical uses.
Very cool. A couple more questions from our audience. Is this technology comparable to linker technology, and does it have the ability to cross the blood-brain barrier? That's a great question.
Yeah, so the short answer is yes. You can modify and load an exosome. So you can program it for different locations, and they do cross the blood-brain barrier. And so there's a number of really interesting applications looking at delivering CNS products to the brain and to specific locations.
Awesome. And one final question from another physician in our audience tonight. Tell us a little bit about your IP. How protected do you feel about your kind of secret sauce, your reagent box in regard to competitors?
Yeah, that's a great question. Again, this field, the immunomagnetic bead core technology has been around for 40 years. What Clara has really innovated is two things that we have IP around. One is we have a custom bead that's actually designed for exosomes, and it actually is able to capture a lot more exosomes to the surface based on that geometry. The second aspect is that release phase. And so it's very difficult to get these exosomes off. And so we have IP around that release phase. And then there's a third part, which I won't really go into here, but it's around downstream microfluidics that allow us to do automated processing in the future.
Great. Jim, thank you so much for being here tonight. Thank you for telling us all about Clara Biotech. This is some really exciting technology. And as you mentioned, I think at the forefront of what's happening in healthcare.
Can I say one last thing on the poll, Katie?
Sure.
I would just like to say, I'm fortunate to be in the room right now with a lot of smart, connected people. And, most of you might not be super familiar with exosomes yet, but you probably know people that are in areas of development and research that this technology could play a part in. And what we're really looking for are high-level qualified introductions to researchers, cancer center labs, diagnostic developers, and others. So if this isn't your space, you probably know oncologists and thought leaders who might be interested, and we'd love to connect and get an introduction if you are willing and able.
To that end, we're going to move on to our next presenter. So we have Lena Biosciences, who is the CEO of Lena Biosciences. And again, she's going to tell us about another very exciting technology. Go ahead, Yelena, and share your slides.
Thank you, Katie. And you got it there. You were able to say the last name. Okay. So, hello, I'm Yelena Vukasinovic with Lenas Biosciences. We have a commercial technology which replicates human body for systematic drug testing. We are positioning the platform for cancer prognostics to optimize therapies for patients using their own tumor tissue after surgery and ultimately biopsy. We are seeking partnerships with oncologists to demonstrate the value of our lab-ready product in a clinical setting. While the precision medicines market is forecasted to grow to $86 billion by 2025, these drugs can only be used with 5% of patients. And for many drugs, the improvement in survival is measured in months. Why? Precision drugs focus on specific mutation. The problem is that cancer evolves past that mutation, and it evolves differently in each patient. Our perfused organ panel is the solution to identify drugs which work for the patient's own tumor. In our platform, we can grow mini tumors from a patient's surgical resection to identify drugs which kill the patient's tumor en bloc. In our platform, we can save patients 4 to 6 months of time to obtain a personalized drug therapy result at half the cost of growing their tumors in mice. In addition, we can test all drugs, not just those that work in mice, to see what works best for the patient, not the mouse. Here are the features of our market disruptive technology, which can select the best drugs for each individual patient. First, our system grows tumor in 3D. This restores cell shape, receptors, which are drug targets, and cell functions such as invasion and metastasis that only exist in 3D. Second, our system provides perfusion because most tumors bigger than 1 to 2 millimeters are perfused. Third, in-well perfusion concentrates cellular secretome, metabolome, extracellular vesicles such as exosomes, cell-free DNA, and evDNA for therapy monitoring and ultimately prognosis. Fourth, perfusion enables intratumor delivery of monoclonal antibodies and antibody-drug conjugates that can only be delivered and tested with systemic blood circulation, which our platform mimics. Fifth, our synthetic hemoglobin blood substitute provides blood-like oxygen delivery for long-term support of immune cells for immune and cell therapy testing. Thanks to the blood substitute, the function of vital cellular enzymes is restored. As a result, in our platform, cellular drug metabolism is increased tenfold compared to cells in a dish to see if the drug really works or it doesn't. In sum, we have a comprehensive test system which can identify drugs that kill the entire tumor, not just the parts of it. And this includes pre-metastatic cells, which eventually make it into the blood. Here's how our system predicts the efficacy of cancer drugs. We have 2 examples here, a small molecule inhibitor and a large molecule monoclonal antibody, Herceptin. These drugs work in 3D, but not in 2D. When tumors are grown in our system, receptors are restored, and drugs work at typical doses we see in clinic. Here is another example. For antibody-drug conjugates, our system can answer if linked toxins can selectively target tumor drug metabolism and eradicate cancer, or provide reactive metabolites that inside the cancer cell create overwhelming oxidative stress and kill it. We can answer this because our system increases drug metabolism log-fold compared to cells in a dish. Next, we can answer if targeting cellular antioxidant enzymes can eradicate future metastasis because our system supports pre-metastatic cells that substantially rely on oxygen. Lastly, because the cells in our system are in 3D, we can tell which molecules cause lipid peroxidation or enough membrane damage that leads to endocytosis of efflux transporters that are responsible for drug resistance to primary therapy. Our team has over 60 years of combined experience in tissue engineering and life sciences. I have 3 issued patents and am the principal investigator on small business innovation grant awards from the National Institute of Health and the National Science Foundation, which funded our technology developments. Our CSO is an experienced biomedical engineer with prior appointments at Emory. Our strategic partner is Specicare, a tumor tissue bank, which we have been using for the initial validations. With Dr. Dixon, who is the chief medical officer at Specicare, we founded a small personalized therapy pioneer consortium comprising small companies and universities. Our technology developments have been funded by prestigious SBIR awards after passing rigorous scientific review, which speaks to our credibility as a company. We developed our own intellectual property and secured the patents at scarce resources. This speaks to our innovation and resilience. We have patented products and customers in the research market, including cancer research and pharmaceutical companies. Our customers are publishing in peer-reviewed publications using our products. The papers that we publish have thousands of reads. We are a part of the ATDC and JLabs incubators. To summarize, through our innovations, we are driving a new market in personalized medicines. We're looking for strategic investors and partners who share our vision for the future of cancer care. We have a solid track record and are in the fundraising mode. The funds will be used for business development, sales and marketing, 510(k) regulatory device approval, CLIA, and related certifications. We ask for your help to secure partnerships with oncologists and healthcare systems. Lena Biosciences, the future of personalized medicines.
Yelena, this is so exciting. Thank you so much for sharing with us. It seems like this could radically change the way we make decisions about cancer treatment. Talk about how this fits into the clinical workflow.
So our device fits nicely into a routine clinical lab workflow. Any clinical lab has standard cell culture and readout equipment to grow and interrogate cells both quantitatively and qualitatively. And our disposable plates have a standard multi-well plate format, which is the most frequently used consumable in a cell culture lab. The only difference is just the attachment of one tube and the placement of the pump outside the incubator. For chemosensitivity, we use a standard cell titer glow ATP assay, which most technicians use, and we can recommend how to multiplex assays, for example, for fluorescence and luminescence to get additional readouts.
Great. We have many questions in our audience here, so I'm going to go directly to those. Two of them are about other, I would say, competitors or comparisons. How do you compare your solution with organoid-based drug sensitivity testing? And do you consider your technology a major advance over tissue/organ-on-a-chip type technologies?
So we're actually at the intersection of both. So our device technology, yes. So we are funded by that NCATS initiative for organ-on-a-chip technologies, but we have a few major differentiators. So for organoids on their own, they're not perfused. So we use that technology in a slightly different way. You can still call it an organoid. And in terms of organs on a chip, we just turned that into a high throughput device. So rather than having, just imagine you want to test 96 cultures in a plate, right? So what you need is 96 tubes coming in, 96 tubes coming out. You need 96 pumps or pump channels. It gets really, really complicated. We boil it down to something really simple when there is just one tube, okay? You still have 96 statistically independent conditions. Just because we have that blood substitute that is really awesome, and it's a special chemical that acts like a hemoglobin in terms of delivering oxygen and taking the CO2, yet it's totally inert and doesn't interact with anything in the medium, so we're just basically using it as a little pump. In addition, with organs on a chip, you get a lot of, I would say, a lot of air in the system. It's really, really small, and it clogs. You can't really do it for a long time. We come from that world and had a technology like that. We thought that it was not ready for the market, not throughput scalable, didn't fit into routine, and we figured this is better.
All right, so a couple of questions about this microenvironment that you are creating for these biopsy samples. So clearly maintaining that microenvironment is essential to accuracy of testing. I want you to be able to talk a little bit more again, how you are keeping that physiologic environment where you want it to be. And then a follow-up question to that is, how do you know, in fact, that your blood surrogate is in fact comparable to actual human blood?
Okay, so one thing, so just to say on the side, like how we actually do it and how we are maintaining. So we keep the cells in 3D. So we have a special procedures where we come from tissue engineering. So we can create spheroids, tumoroids, anything. We can isolate the cells, mix and match them. We've done this for years. We take this a step further, in effect, where we try to keep the original heterogeneity of cell types. So the method itself is similar to what Calvin Coe has at Stanford, but we have the perfusion, and we're not culturing at the air-liquid interface, because that it doesn't have the physiological exposure. It's super physiological oxygen, right? So we're trying to keep that immune stroma. We try to keep all the cells on block. Yet we keep them submerged, but we have the blood substitute. How do we know? Well, we measured actually oxygen consumption rates in the cells. And we do know that even if we had a hepatoma using the appropriate medium, we can increase the oxygen consumption rate 4 times. So going from cells being completely glycolytic to actually cells utilizing mitochondria. And with that, we were able to go approximately 60% of the lower end of the primary human hepatocyte respiratory metabolism. So we do know that, and we measured, like I said, oxygen, how does it work? And these compounds, these similar compounds have been used actually as blood substitutes. The difference, our difference is that we're not making emulsions. So we don't need huge equipment to supercharge them with oxygen. We just equilibrated. That's covered by the patents. So they always have the exposure, the right exposure to oxygen. Important thing to remember, because the tissue is submerged, right? And it's always perfused in the media. You cannot give it more oxygen than it can take from the environment. And it's the tissue, it's that gradient between the tissue and the oxygen that actually drives it inside. So it's almost like an oxygen hemoglobin dissociation curve. Ours is a little bit more linear, but we have a buffer between the tissue and the blood substitute, which is that medium that makes it just perfect.
And again, the idea being the more lifelike you keep the environment, you're going to be able to get these results. They're going to be more accurate, and you're going to be able to get them much quicker, correct?
Yes, absolutely. So our goal, so if you really create the right environment to keep the tissue, because some of these liquids have also been used in the past for like to maintain organ transplants. So if you really create an environment that the tissue likes, you're actually able to test drugs a lot earlier. And if you miniaturize that, you don't have to wait too long for anything to grow in an animal. You just do it right on spot. So our goal is, with the further validations, to really make a big leap, get a tissue, we don't think that, mutations, if you culture for a short period of time, they're not very likely to happen because they happen more as the cells divide. But what we're really trying to do, this is going to be our next big thing, is to demonstrate that the function has not changed. Then you have a perfect replica. Then you have a tumor in a dish. So, and for that, we will be working with SpeciCare most likely to get the gene expression as it comes out of the patient. When we put it during transport, when we get it in like a day or two in our system, and if we can show that it hasn't changed, boom, that's really huge.
Well, this is super exciting, and I know will be, like I said, transformational for cancer treatment everywhere. So thank you so much for being here. I know my wheels are spinning about what other potential applications this might have, and I am going to have to hold off until our networking after this because we're going to move on to your poll. Our last but not least, of course, our third presenter of the evening is Andy Olson. He is the co-founder and CEO of Lena Biosciences. Andy, go ahead and share your slides. We are eagerly awaiting our last pitch. Like I said, 3 excellent companies this evening.
Thank you, Katie. I'm Andy Olson, co-founder and CEO of Lena Biosciences, and we're an emerging molecular diagnostics company. We're solving a problem with infertility. Why infertility? Well, there's an increasing trend as couples wait longer to have children, and 1 in 8 couples experience infertility. So the chances are that you probably know someone who has experienced infertility and infertility treatment. And if you do, then you know that the focus is primarily on the female partner. Yet 50% of infertility is due to male factors. And the current semen analysis, looking at sperm with a microscope, counting how many are there and counting how many are swimming, is a poor diagnostic test with about 15% sensitivity. This leads to a lot of unexplained infertility. 65% of couples drop out of treatment due to the emotional toll. And there's 3 times more divorces, which is just tragic. So we need a better test that identifies male infertility and guides treatment to a procedure that's going to give the highest likelihood of success. We can increase the diagnostic power of semen analysis by analyzing sperm DNA, and specifically epigenetics. Epi is a prefix that means on, and genetics is the DNA. You can see in this cartoon over here, on the DNA are biochemical modifiers that turn on and off gene expression. One of those is DNA methylation. The DNA is packed tightly into the head of the sperm. And so what we do is break open the sperm, isolate the DNA. We convert the DNA where there's an absence of these methylation marks. And then we do what's called methyl-seq, and it's next generation sequencing. And that allows us to then report results based on the methylation of the DNA that correlates with the condition of infertility. So how do we know that this works? Well, 2 of our clinical and scientific advisors have published papers in this area showing that DNA methylation predicts male fertility status and embryo quality, and also things like therapeutic responsiveness based on methylation of follicle stimulating hormone, also known as FSH, one of the standard treatments. We have very robust IP in this area with worldwide exclusive licenses to the IP, with broad claims for methods of using DNA methylation in this way and the machine learning and artificial intelligence algorithms behind it. Over the past 2 years, we've completed validation studies showing that our DNA methylation biomarker is enriched in infertile patients compared to the fertile donors. And you see in this figure of box plots an increase in epigenetic biomarker enrichment in the infertile population of a prospective study, as well as our retrospective study, compared to the fertile sperm donors. And most importantly, we see our biomarker is more prevalent in men with normal semen analysis. So in a study of 112 couples going through infertility care, 90 men were considered fertile based on the standard semen analysis. And we found our DNA methylation biomarker identifying male factor infertility in 36% of these men. So we've developed an epigenetic sperm quality test with a positive predictive value of 97% and a negative predictive value of 96%. We did these studies with partners at the University of Utah and Baylor College of Medicine, along with our clinical laboratory partners, one of which is Predictive Technology Group. We signed a strategic collaboration and commercialization agreement with them, where it's a revenue sharing agreement. They have a female infertility test, and so our male infertility test goes very nicely with that and gives us a channel partner directly into this market. In addition, we have developed an at-home collection kit so that men can receive a specimen collection kit at their home, collect the specimen at home in the privacy of their own home, and then send it into the laboratory for analysis. We have 3 ongoing IRB-approved studies with Donor Egg Bank USA, Eugen, Barcelona, the largest infertility clinic in Europe, and then Boston IVF, the largest infertility clinic in the US. There's other indications in our pipeline, but we're focused right now completely on fertility. And we foresee in the market significant opportunities for exits. In the direct-to-consumer market, there's a number of companies focused on male reproductive health. And we think that the opportunity will actually be driven more, it'll drive more value in the market with the clinical use and going to these fertility clinics, the laboratories that serve clinics that are doing fertility testing, and then also the pharmaceutical companies that provide reproductive medicines like Ferring Pharmaceuticals and EMD Serrano. And then there's also the opportunity for an IPO. We anticipate the first exit opportunity as early as next year, the latter part of next year, before we internalize laboratory operations. And then another exit opportunity potentially after we launch our second test using this epigenetic information. We have the team to do this. So my co-founder and I have prior startup experience. I've led commercialization in 5 biotech startups with 4 exits. And Kristen Brogard, my co-founder, has a PhD in the field of epigenetics. We've assembled a team of talented technologists, clinicians, former operators in this space, and pioneering scientists. And our team uses what's inherent in the biology to remove the guesswork and the trial and error in infertility care. We're looking for feedback, potential partners to bring this technology to the market, and also potential clinical pilots. Thanks, and I look forward to your questions.
Andy, thank you so much. Oh, I love the emojis. Here they come. You know, this is exciting because, as you mentioned, there are many, many couples out there that are struggling with this infertility issue. And as you mentioned, most of that, emphasis is on the female, the mom to be, as opposed to the dad to be. So this is very exciting because clearly there is a need for something like this. One of our audience members actually stole one of my questions, so we'll start with that. Dr. Kona says, epigenetics with AI is very exciting and has broad application. Have you thought about broadening this to other diseases with significant epigenetic impact, like obesity, psychiatric illnesses, etc.?
Yes, absolutely. Thanks for the question. So, I mean, there's a clue that we found when there appears to be a gender disparity in the disease. That typically is an indication that there's something going on epigenetically. So, for example, with autism, it's mostly in boys. And with autoimmune disease, things like rheumatoid arthritis, my mother suffers from rheumatoid arthritis, and it's mostly in women. And so that's a clue that there's possibly something going on epigenetically in there. We've licensed biomarkers from some of our academic collaborators in the area of autism and autoimmune disease in particular. And absolutely, obesity, there's a number of things in the pipeline with our academic collaborators that we're focused on potentially in the future applying this technology as well.
Well, and a follow-up question to that, are you planning or thinking about separating the company to focus on fertility while pursuing testing for these other indications?
Yeah, so one of the things we've thought about is setting up subsidiaries underneath our corporate entity and doing that and either licensing things out to them or to partners that can fully commercialize it, or setting up a focused team in a subsidiary to commercialize the technology. We think some of these areas are very attractive and we're leaning more towards setting up teams with some of them. Others, it makes more sense to out-license the technology and let others do that commercialization and then either generate royalty revenue or a revenue sharing kind of arrangement.
Sure. So I have a couple more clinically related questions. You mentioned you have a large upcoming clinical study. Can you tell us more about that?
Yeah, so we just got the data actually for 1,500 blinded samples where we're doing the analysis on those and then comparing it to the outcomes. And this has actual live birth outcomes. And that's what a lot of the clinicians that we've been working with have asked for is, how does this translate into live birth outcomes? So we're just analyzing that data. We intend to publish that here hopefully in the journal Fertility and Sterility, which is the primary journal that the reproductive endocrinologists and reproductive urologists read. So that's a big pivotal study that we're getting out. And then we have these 3 IRB approved studies that are focused on sperm that failed egg donor cycles. And these are egg donor cycles where we know that the eggs are good. They come from lots that have had success in other cycles with other sperm. And so now we're getting the sperm that failed those egg donor cycles with a built-in control. It's going to be phenomenal data, really controlling for the female factor and identifying that this is real and that there's an issue with the male factor that can be attributed to the failure of that cycle. And you can identify that up front, then you can avoid a lot of the unnecessary treatment that's not effective and go to the procedures that are effective when there's a male factor issue.
Certainly. Speaking of that, you talked about the ability to predict, utilizing this information and data to predict whether FSH was going to be a useful treatment or not. I'm assuming that's what you're talking about with other types of procedures and/or treatments. Is that true?
Yeah, so a lot of the treatments start off with ovulation induction, hormone shots for the female to induce additional ovulation, and then so timed intercourse using that ovulation induction, and then also artificial insemination, or also known as IUI, intrauterine insemination. So what we've shown in our preliminary data is that we can predict success or failure of those based on the sperm epigenetics, and then also potentially miscarriage. So you can often get a pregnancy, but it's really, can we keep the couple pregnant and not have a miscarriage? And so we're seeing that we can be predictive of miscarriage as well. So getting to that live birth outcome that all the couples and the physicians want to achieve.
That's fantastic. Again, I know a struggle for many companies. We really appreciate you being here tonight, Andy, and telling us all about Inherent Biosciences. I can definitely see many applications of this, not only in infertility, but outside of that. So very exciting.
Thank you.
We are going to go ahead and put up the poll for Inherent Biosciences. And again, these CEOs and founders really appreciate your feedback. There's a link in the chat, so you can go ahead, please, and give your feedback. Again, we are trying to beat the very first poll numbers we got of the evening, so 60 plus. Please vote. And while you're doing that, I am going to talk a little bit about what comes up next. Oh, yes. Andy will also talk to you in the end in our networking session, along with our other CEOs, about really what it is that they want from our network. So please join us, because again, that's what happens next. Once we close down the live pitches, we would love to see you in our networking lounge so you can continue the conversation and continue to ask more questions. I know there were several questions within our Q&A that we did not make it to tonight. So please come join us, because these folks are very passionate about what they are doing, and I know they cannot wait to answer more questions from you.